The overall goal of this procedure is to measure chemotaxis in an automated, real-time assay which utilizes electric cell substrate impedance sensing, or ECIS. This is accomplished by first preparing the ECIS Taxis electrodes by filling each chamber with 0.5% agarose in complete RPMI medium. The next step is to simultaneously cut 2 wells into the agarose of each chamber with a 14-gauge cannula jig and to aspirate out the 2 agarose plugs. In each chamber, one well is filled with a single-cell suspension, while the other is filled with chemoattractant. Next, the electrode is connected to the ECIS theta system through the array holder that is placed in an incubator set to atmospheric parameters appropriate to the cell type being tested. Ultimately, cellular chemotaxis can be observed through measuring the change in resistance in real time as cells reach the target electrode between the 2 wells.
The main advantage of the ECIS Taxis assay over existing methods such as Boyden chamber assay or conventional microscope-based under agarose assays is that it allows for real-time automated measurements of chemotaxis without the need for ongoing manipulation of the system.
To begin electrode preparation, stabilize the gold surface of the ECIS Taxis electrode array by pretreatment with sterile 10 millimolar cysteine in deionized water for 15 minutes at room temperature under sterile conditions. Following pretreatment, aspirate the cysteine solution from each electrode chamber. Rinse 3 times with sterile deionized water and replace with 250 microliters of complete